thank you for the organizers for inviting me for this to present here. I'm going to talk about a bunch of stories about that we've tried to identify using genomic data to understand lipid biology. So a lot of this talk will really use snippets of what came before this from Ramesh's talk and from Neela's talk. So I'm a population geneticist, and so I tend to sort of classify genetic variation in my head in these 4 categories. I divide this by allele frequency, so common versus rare variation, or by genomic context, right? Is it coding variation? Does it lie in the exome, or is it non-coding? And depending on the question you're trying to answer, you're going to use very different approaches to identifying variants in these different classes. The particular class of questions I'm interested in this work is the non-coding variation that's relatively common in the population, and genome-wide association studies are the class of studies we use for this, for these kinds of questions. So very briefly, a genome-wide association study is this very large population-based study where you work with individuals with a spectrum of phenotypes, genotype each individual at about a few million positions in the genome, and then test each of these million positions, or actually few million now, to see if, which of them are associated with a phenotype, right? That's what we're looking for, finding positions in the genome, common positions in the genome that are associated with your phenotype of interest. The class of phenotypes that we were interested in were related to lipid levels, and these are probably things you've heard of. Cholesterol, HDL is sort of, or high density lipoprotein is what we often think of as a good cholesterol, right? LDL is the bad cholesterol, which we associate with increased risk of heart attacks, plaques in arteries. There's also triglycerides, which I will refer to from now on as TG. And there's total cholesterol, which is the sum of all of these 4. So what I'll talk about are the results of these genome-wide association studies that we perform on all of these 4 classes of lipid molecules, and also what we call non-HDL, which is all of the cholesterol molecules that don't include HDL. So lipid GWAS has really actually been at the forefront of human genetic studies for the last few decades. We started with about 10,000 samples, all European, and they found 18 positions in the genome associated with lipid levels. And if you go all the way to the bottom, in 2018, that had moved up to 300,000 samples. And we discovered about 250 positions in the genome associated with lipid levels, right? So the study I'm talking about was published in 2021, part of the Global Lipids Consortium, where there was a huge bump in the sample size, thanks largely to the UK Biobank, where we had 1.65 million samples, still mostly European, but this was the largest study on lipid levels in non-Europeans, even though that number is still very tiny. This was a meta-analysis, so included 201 primary studies on these 5 lipid phenotypes that I spoke about. So I'm not going to speak about this GWAS, but what I'm going to speak about is what a GWAS can't do, right? So a GWAS, what it gives you is these multiple independent signals in the genome associated with lipid levels. So here you have these peaks in the plot. This is called a Manhattan plot, and on the x-axis is sort of genomic position, and each dot here represents each of the few million positions that were tested for association, right? And on the y-axis is the minus log 10 of the p-value, so the higher up you go, the more significant you go. Anything above the red line over here represents genome-wide significance. All this is saying is that we have a lot of positions of the genome that we think influence lipid levels. That's what a GWAS tells us. What it doesn't tell us is, so what? Right? What do we do once we have these thousands of signals associated with lipids? And that was the kind of question that I was interested in asking. The reason a GWAS doesn't tell us that biological information is because of the nature of genetic information. So as Ramesh, I think, alluded to earlier, your genetic data or your genetic material travels in blocks, which means that if you see a significant association at a particular point of the position in the genome, you're not entirely sure if it's at that position, if the causal variant is really that position or some other variant in the vicinity of that region. Right? This is because of something called linkage disequilibrium, where essentially variants don't move independently of each other. Variants that are close by tend to be inherited together. Right? So we don't often know what the causal variant at each of these peaks is. What we also don't know is what is the gene mediating this association. I mean, you would assume that you have a variant associated with lipid levels, but there must be a gene somewhere in between that's doing the work, that's mediating this association. And a GWAS doesn't automatically tell you what that gene is. Answering these is important. Again, knowing the causal gene helps us understand the biological mechanism of these phenotypes we're studying. And again, as Ramesh said, there are these studies showing that genes with genetic support are twice as likely to be approved in clinical trials. So understanding the causal gene is a particularly important result that we want from a GWAS. Why should we also know the causal variant? It's because, again, of polygenic risk scores. If we want to use genetics to predict a person's genetic risk, we need to know what exactly the causal variant is that is responsible for that association. Otherwise, these risk scores are unlikely to represent truth. So this is really a huge problem in the field, moving from association to biological function. Biologists are not very fond of people doing these genome-wide association studies because we sort of dump all of these genes and signals on them, and performing the experiments in the lab, validating each of these thousands of associations one by one is not easy. In fact, if you look at the graph on the right, you see that the number of genome-wide association studies has increased very quickly, but the number of functional studies, shown in orange, has really not kept pace, which means that we have these thousands of associations for pretty much every phenotype that you can think of has been studied. We have these associations, but very often we don't quite know what to do with them or what they mean biologically. And this is not a novel problem. This is something that many people have tried to think about and is actually a very important challenge in the field. But what we tried to do was use a very simple approach where we tried to incorporate functional data into genetic data to try and arrive at the causal gene and the causal variant. So what we did was we started out with these 1,150 independent signals that were associated with lipid levels, and at each signal, and here I define the credible set as the set of potential variants that could be causal at a signal, right? We don't know what the causal variant is, so I'm saying any variant that has a 95% posterior probability of being causal, I'm going to call that the credible set. What we then do is integrate that this genetic information with functional genomics data, for which we have a lot more of now. We integrated with eQTLs, and I'll talk about that a little bit. These are variants that affect gene expression. We also integrate these with what's called promoter capture-C, which try to capture the three-dimensional looping of DNA in the nucleus, right? If you're a geneticist, you sometimes tend to think of DNA as completely linear because all you see are these FASTA files scrolling by on your screen, but in the nucleus, these are 3D physical structures that are looping together. And what these promoter capture-C experiments try to capture are which parts of the genome are close together physically. We integrate these also with other functional openly available functional genomics data, including OpenChromatin and ChIP-seq, and try to arrive at biological insights. I won't talk about all of this, primarily because I had forgotten this was a 30-minute talk and I had prepared for a 60-minute one. But I will give you maybe a couple of anecdotes about what we found. Okay, so we first use eQTLs, incorporate eQTLs into genetic data. What are eQTLs? This is really a fancy name to a very simple concept. An eQTL is a variant that's usually near a gene, within a megabase of a gene, that affects the expression of this gene in some way. So statistically, what you would expect what an eQTL would look like is like the box plot below, where an individual carrying different genotypes of this variant would have different levels of gene expression of this gene. Right? That's what eQTLs are. And what we're looking for are, at a given position in the genome, do the GWAS signal, so again, this is that Manhattan plot, zoomed into one window, does a GWAS signal and an eQTL signal sort of look similar? Do they show the same patterns of association? Okay. And what we use for this is this massive publicly available eQTL data set called GTEx, which has eQTL data from humans from 49 different tissues. And what we did was integrate our GWAS data for these 5 phenotypes with these eQTL data sets from GTEx and found those GWAS associations that co-localize. What we see is that out of 1,750 genes, more than 1,000 actually co-localized. So these are the potential causal genes at those 1,750 GWAS associations. And what we see is using EQTLs alone still doesn't always help you get at the causal gene per locus. Per GWAS locus, we still can narrow in on anywhere between 1 and 16 genes. So we actually need more kinds of information to arrive at a single causal gene, if there exists one. The figure on the right shows an example of a colocalization at one novel gene that we found, RRBP1. The top plot is the GWAS association in this region. So this represents an approximately 2 MB window of the genome. You see a very strong GWAS signal for LDL levels. And the bottom plot shows that same Manhattan plot for EQTLs in the liver. So this is the EQTLs for RRBP1, where you see that same peak of significance, colocalizing very nicely with that LDL signal, really pointing to this as the likely causal gene there. Okay. What we also see is we took these 1,076 colocalized genes and looked for whether they were present in other known data sets, what we call silver standard data sets, because again, it's very hard to know what a gold standard association is, but if you look at other data sets that include genes associated with severe cases of lipid disorders, like lipidemias, we see that our 1,000 or so genes are significantly enriched in all of these mostly independent data sets. And this is really interesting because it shows that a GWAS captures small effects of variants, leading to small effects on the phenotype. These other databases capture knockouts, gene knockouts, so large effects of, like complete knockouts of genes, leading to large effects on phenotype. And both of these sets of genes converge upon the same set of genes. This means that even though we're studying very tiny effects of genetic variants on phenotypes, these have information about those rare diseases, even though we're not directly studying them. There is a convergence of evidence going on there. Another question that we were interested in, and this really segues very nicely from the previous talk, is trying to identify transcription factors that could have an outsized effect in lipid biology. Are there certain transcription factors that, for some reason, regulate more lipid-related genes than not? And to do this, we use this method called stratified LD score regression. Just conceptually, what that does is it takes your GWAS signal across the genome, it takes your summary statistics, it overlays that with ChIP-seq data, with publicly available ChIP-seq data for 161 transcription factors, and what it tries to ask is, do your most significant signals tend to overlap with the most significant ChIP-seq peaks more often than by chance? That's sort of intuitively what this method does. And when we applied these to our 5 GWASs, we found 8 transcription factors significant across all 5 phenotypes. This is a highly conservative set of transcription factors. If you're a lipid biologist here, a lot of these are familiar to you. These are some of these are smoking guns that you would expect to see. But what we were particularly interested by was this one transcription factor, RXRA. Now this was a gene, this is a retinoid X receptor alpha. This is a gene that was previously implicated as a GWAS locus. So we had known that there was a GWAS signal at this gene even before we did this study. But now what we we've been able to make this arrow, split it up into more arrows, where we have a little more resolution, where we think it affects LDL not directly, but by acting as a transcription factor on a bunch of other genes that mediate its effect. And this sort of ties to this paper that came out in 2018 that spoke about the omnigenic model of complex traits, where we sort of start thinking about traits as being Mendelian, and then we talk about them as being complex, but what actually is going on in the cell that allows all of these different genes and variants to collectively contribute to a phenotype. And the authors here propose this model where your phenotype is likely to be influenced by a bunch of core genes that have a direct connection to the phenotype, and perhaps these core genes are the ones most likely that are found in rare diseases. But you also have a bunch of peripheral genes that indirectly affect the phenotype by sort of acting on some intermediate genes, which in turn act on other genes and so on and so forth, till you have a change in phenotype. So this is what has been called the omnigenic model, and transcription factors like RXRA is likely one of those peripheral genes that acts on LDL levels by by sort of by acting on all of these intermediate genes, all of which have also been found in our colocalization, EQTL colocalization signals. How much time do I have? Oh. Okay, all right. So what we then tried to do was, I spoke about trying to arrive at causal genes, but I still haven't spoken about trying to find the causal variant. And so we used a very simple framework just to see, can we try and prioritize which of these variants is likely to be causal. And to this, we started from first principles. So we said, okay, what would a causal variant look like? What would a functional variant in the genome look like? You'd expect it to perhaps regulate gene expression. You'd expect it to physically interact with the gene. You'd expect it to lie in open chromatin, which is a proxy for activity of the genome. And you would also expect it to have active genome marks, active histone marks. So there are histone marks that we know are active versus repressive. So we started out with this very basic framework of rules, and then built a more quantitative framework out of it, where we said, okay, for each of the variants in all of our credible sets, we annotate them with EQTLs, promoter capture C, histone modifications, and also what's called a regulome DB score, which is a machine learning based estimator of that gives you a likelihood of how functional a variant is likely to be. And we prioritize variants that have essentially that are likely to be causal based on at least 2 out of these 4 metrics. And the reason we use at least 2 out of 4 is because unlike genomic data, functional genomics data sets are still lagging behind in terms of how much diversity they capture. We still have very few functional genomics data sets in terms of sample size and in terms of developmental context. We know that these functional annotations are likely to differ in different tissues, not just in different tissues, but also in different stages of development. And there's no way a single functional genomics data set can capture all of that. So we tend to have to be more relaxed when we make rules that capture information from functional genomics. What we get from these is a sort of 14 loci with a single prioritized variant, many more with anywhere between 2 and 10 prioritized variants that we have up on a browser for anyone to download and look at. Now, I'd like to present maybe one example of a gene that we annotated using this framework. This is CREB-RF, which was a regulatory factor. What you see here is a GWAS association of CREB-RF with HDL levels. What we also see right away is an EQTL colocalization in adipose. So this is also interesting because HDL is known to be more adipose specific, whereas LDL levels are more liver specific. So it's nice that we saw an EQTL in adipose, which is likely to be the tissue actually of interest here. And what we see here is this is a lot going on here, but what you actually are seeing is that a zoomed in version of the top two panels, where we see a physical interaction in adipose and the liver actually between a variant in that credible set and the promoter of the gene. So here. We also add annotations from open chromatin, where we also see that the variant is likely to lie in open regions of the genome. And we also see that this variant is likely to be a promoter in the liver actually, not the adipose. And this again points to not to any tissue, a given tissue's data set not being perfect. So we sometimes have to take information from a different tissue because you might not have complete information for a given data set. Okay, so we used one approach, which is functional genomics, to try and do what's called fine mapping, which is finding the causal variant from this credible set of variants. But another approach that's been used is called using multi-ancestry data sets, where instead of using Europeans only, you use information from Africans or from South Asians or from East Asians, all of which actually reduce your the size of the credible set that at a particular locus. And this is because of LD, because especially using African populations, the LD is so much smaller, these linkage disequilibrium blocks are so much smaller that your credible sets tend to go down. So what we wanted to ask was, if we perform our functional fine mapping on European credible sets, compare it to fine mapping using multi-ancestry data, are these two likely to converge on the same set of variants? And they do. So if we, okay. If we compare the variants found from functional fine mapping to those using multi-ancestry GWAS, we see close to a four-fold enrichment, showing that these are two complementary approaches to arrive at the causal variant, and both of them can be used in different situations, but they tend to converge in the same set of variants, which is a good sign. I'll maybe end with one final anecdote of the kind of story we found from this study, where we not just looked for for GWAS signals in the entire data set, but in males and females separately, and tried to see how many sex-specific signals do we see. Well, spoiler alert, most signals are not sex-specific. Most signals out of those 1,000 were actually common to males and females, but there were 64 loci showing significant sex-specific effects, out of which 10 of them had an associated sex-biased EQTL. I'm going to show you one of them. So this is a gene called UGT2B17. This shows a GWAS signal for LDL for this gene, well, at this locus. And in females, the same plot for the same y-axis, you see no signal at all. This is controlling for sample size, so it's not that sample size is the main reason for that lack of signal there. What do we see at EQTLs for this gene? We again see the same sort of signal replicated, where again you see a very strong liver EQTL for UGT2B17 in males and none in females. So now we think it's a sex-specific EQTL that is mediating the sex-specific GWAS signal, which we think is very interesting. What makes it a little more interesting is that if you go and look a little more into UGT2B17, this is a gene that acts upon testosterone. It metabolizes testosterone. So there's a very clear biological connection between this genetic signal and what you'd expect to be a very sex-specific phenotype. It gets even more interesting because if you've looked at Manhattan plots, I'll say this very briefly, this is a very weird shape for a Manhattan plot. We usually just see peaks. We don't see these plateaus. So we try to figure out what's going on there. Why do we see that plateau? And what we see is that there is actually a common copy number variant at that locus. These are not single nucleotide variants, but in actual deletions or duplications. What we see is that about 30% of people have a copy number variant right at that locus, and it's the copy number variant that's actually capturing this GWAS signal. If we condition on that CNV, the EQTL effects go down significantly. So it's actually a duplication in that gene that increases gene expression, but for some reason, that duplication only increases gene expression in males, but not in females. And we don't know why yet, and that's a story that I hope we'll find answers to at some point. So I'll stop here, but I'd like to thank all of the contributing cohorts. Such studies are really possible because of an immense amount of data sharing, and most of the South Asian populations came from Dwaipa and Bharadwaj in India, but also the UK Biobank, and this was led by my advisor, Casey Brown. And I'll be happy to take any questions.
Hi, ma'am. It was an interesting talk. Actually, you told you have been using GTEx data. So GTEx is a non-disease population, the samples are from non-disease population. So how do you associate it with the GWAS?
Yeah. So all of the GWAS is also from a non-diseased population, right? So we looked at individuals who have a range of cholesterol levels, but not beyond the spectrum of what you would call having a disease. And these are individuals with fairly healthy levels of cholesterol levels. And GTEx also, yes, is mostly a healthy group of people. But I think it's both a challenge and an advantage of common disease genetics, or common variant genetics, where these variants will also act the same way in a healthy population. It's not a single variant that will make you diseased or that will give you the disease. It's a collection of a bunch of variants that will push you beyond that boundary of, let's say, having cholesterolemia. So you would still find these effects even in a healthy population. Yeah.
Thank you.
Ma'am, I want to know that is it possible to identify causal genes using candidate gene approach? Like if we have limitations of other kind of data available, but we just have information on genotypic information.
So I think the candidate gene approach is tricky right now because I think there have been too many, we've been burnt too much. We've been burnt way too often with, the first few decades of human genetics were candidate gene approaches. And so many of those associations turned out to be false positives that now I think it would take a candidate gene association itself would not be sufficient to perhaps help, but maybe that along with orthogonal layers of evidence would be sufficient. Does that answer your question? Is that?
Actually, my point is that if we have a set of information on genotypic data, so is it possible to take up some kind of causal, like identification of causal genes? Or is it like we will be needing further information on that?
So you're saying if you only have genetic and phenotype information, will you be able to
Right,
find the
ma'am.
causal gene?
Yes, ma'am.
In certain cases, yes. Right? So for example, one approach you could do is just take the closest gene. You have a locus, take the closest gene, and 70% of the time that will be the causal gene. It's the other 30% of the time that you have to sort of play around with these approaches and see what do we get.
Thank you so much, ma'am.